Okay, let's unpack this. You're navigating a ton of information as a dental student, right?
Absolutely.
You're learning about the oral cavity, the specific bacteria linked to caries and periodontal disease, but the body is this whole complex ecosystem. What if some of the most critical knowledge for treating your patients involves organisms that don't just live in the mouth, but interact with the entire body? Organisms that might even be, cousins to those you've already heard of, capable of causing serious, even life-threatening disease. Maybe by seizing an opportunity when a patient is vulnerable.
That's exactly where we're headed today. We're taking a deep dive specifically tailored for you, pulling the most important nuggets from your provided source material on the diverse and sometimes surprising world of Corynebacterium.
Think of the stack of notes as your personal guide. We're going to work through it together, moving from the broad strokes of this widespread group of bacteria to the specific details that are most relevant to your future in dentistry. Our mission is simple: to help you quickly grasp what's most important from this material by exploring a Corynebacterium, understanding its general characteristics, appreciating its pathogenic potential beyond the most famous species, and zooming in on exactly why its presence, or potential presence, matters in the context of the oral cavity and overall patient health.
Yeah, we really want to give you that detailed understanding you need, focusing on the clinical implications and diagnostic considerations directly applicable to your field.
So let's jump right in and get oriented. Your source material starts by telling us that Corynebacterium isn't just one thing, it's a remarkably diverse genus.
That's right. It's found everywhere, really. We're talking about organisms that can be plant pathogens, animal pathogens, and saprophytes, meaning they live on dead organic matter in the environment.
Saprophytes, okay.
Yeah, so this ubiquity is the first hint that they're a large and adaptable group.
But the ones we're focusing on, of course, are the ones that interact with humans. And the source notes, they are ubiquitous on us too. Part of our normal flora.
Absolutely. They are commonly found as commensal organisms just living peacefully on human skin in the upper respiratory tract, which includes the nose and throat, and in the urogenital tract. They're essentially normal residents in many parts of the body.
So they were just hanging out, mostly harmless.
Many of them are commensals, yeah. And importantly, some non-pathogenic species, often grouped under the informal term diphtheroids because of their appearance, are even part of the indigenous oral flora.
Diphtheroids. Okay, we'll come back to that term.
Definitely. However, and this is the crucial distinction your source highlights repeatedly, while many are harmless, a number of Corynebacterium species are important human pathogens capable of causing significant disease.
And when you hear important human pathogen and Corynebacterium, the first one that probably comes to mind for most people and the one most studied is
Corynebacterium diphtheria. Yeah, this is the classic, historically significant species associated with the disease diphtheria. But as we delve deeper into your source material, you'll see that the story of Corynebacterium in human health extends far beyond just this one organism.
Okay, before we get into what they do, let's spend some time understanding what they look like. Morphology is often the first step in identification, and your source provides a lot of detail here, highlighting that it can also be, well, a bit complex with this group.
It definitely can be. Fundamentally, they are described as gram-positive rods.
Okay.
So under the gram stain, they appear purple, and they are rod-shaped bacteria.
Simple enough on the surface. What about their oxygen requirements and other basic traits?
They are aerobic or facultative anaerobic, meaning they grow best in the presence of oxygen, but they can adapt and grow in environments with limited or no oxygen.
Facultative anaerobic, so that's relevant for, deep tissue infections.
Exactly. Places like abscesses. They're also non-modal. They don't have flagella to swim around, and they are catalase positive, which is a specific biochemical reaction used in the lab to help differentiate them from other groups of bacteria. Got it. Catalase positive. But here's where it gets really interesting with Corynebacterium, and something your source emphasizes. Their appearance is anything but uniform. The term used is pleomorphism. Break that down for us.
Yeah, pleomorphism. It means having variable shapes. These bacteria don't consistently look like perfectly straight, uniform rods. They can appear irregular, sometimes swollen at one end, giving them a club shape, kind of like little drumstick.
Club-shaped, okay.
They can also be curved or tapered, just variable.
And their arrangement after division is quite distinctive.
Yes. Your source describes V-shaped arrangements, which result from cells dividing and remaining attached at an angle.
V-shapes.
But the most classic and visually striking arrangement, particularly noted for C. diphtheria, is described as resembling Chinese letters. or a palisade, like parallel rows of fence posts.
The Chinese letters arrangement, that sounds quite specific. How does that physical arrangement come about?
It's due to a characteristic snapping division during binary fission.
Snapping division. Yeah. When the bacterial cell divides, the two daughter cells don't completely separate and straighten out. Instead, they remain connected at a point and sort of hinge or fold over each other, creating these angular letter-like configurations or stacking up in parallel rows. It's a pretty unique visual clue under the microscope.
Huh. Interesting. So, specific dimensions for the main culprit, C. diphtheria.
The source gives dimensions, typically 3 to 6 micrometers long by 0.6 to 0.8 micrometers wide. And yes, they can have those characteristic swollen ends, which contributes to the club-shaped appearance and is often where you'll see other key features, like the granules.
Granules. Right. And crucially, they are non-sporing.
Correct. Non-sporing. They don't form the tough, resistant endospores that make organisms like Clostridium or Bacillus so difficult to eradicate.
Okay, let's talk about those fascinating granules mentioned in the source. These are a really important microscopic feature, aren't they?
They absolutely are, particularly for identifying C. diphtheriae. Your source highlights reddish, purple granules that can be found within the bacterial cells, often concentrated at those swollen ends we just mentioned.
They have a few different names.
Yes, the source uses metachromatic granules, Babe Ernest's granules, or volutin granules interchangeably. All refer to the same structures.
Metachromatic, Babe Ernest's, volutin. Got it. And chemically, what are they made of?
They are accumulations of polymerized phosphates. Think of them as storage depots, energy reserves, or phosphate reserves the bacteria can draw upon.
Storage depots, okay. And how do these granules affect what you see under the microscope?
Because they stain intensely with certain dyes, they give the bacterial rod a distinctive beaded appearance.
Beaded.
Yeah. Instead of a uniformly colored rod, it looks like a string of brightly stained beads, especially at the ends. This beaded appearance, along with the characteristic arrangements and morphology, provides valuable clues.
And the source specifies particular stains that demonstrate these granules best.
Yes. Loeffler alkaline methylene blue is the classic stain mentioned early on. Later in the diagnostic section, the source also lists Albert, Neisser, and Ponder stains specifically for demonstrating these metachromatic granules.
Albert, Neisser, Ponder.
Okay. Yeah, it really reinforces how important this visual characteristic is for identification in the lab.
Now, we need to talk about diphtheroids. Your source notes that these morphologically resemble diphtheria bacilli, which sounds like a potential trap in identification.
It definitely can be, and that's why the source is careful to differentiate them. While they're gram-positive rods and belong to the broader Corynebacterium genus, diphtheroids, these are the non-C. diphtheria coroniforms, they tend to stain more uniformly under the microscope. They often lack that distinct beaded appearance caused by abundant granules.
So less beady, and their arrangement is different too.
Yes, instead of the pronounced Chinese letter, or angled arrangements typical of C. diphtheriae, diphtheroids are more commonly found in simple V shapes, or in palisades, those parallel rows. Sort of like a picket fence.
Palisades, okay. And the granules?
They have few or no metachromatic granules compared to C. diphtheriae. Much less prominent, if present at all.
So why is this distinction between C. diphtheria and other diphtheroids, based on morphology, so crucial for diagnosis, especially in a clinical setting?
Because you might see gram-positive rods in a smear that look vaguely like Corynebacterium, right? But if they lack the classic beaded appearance from granules and the specific arrangements, they are much more likely to be a harmless commensal diphtheroid from the normal flora, rather than the potentially life-threatening C. diphtheria.
Ah, I see.
Relying solely on basic morphology is unreliable. The presence of granules and the specific arrangement are key visual discriminators that prompt further investigation, while their absence makes C. diphtheriae less likely, shifting the diagnostic focus. Mistaking a commensal for a pathogen, or vice versa. Well, that could have significant consequences for patient management.
That really underscores the importance of those finer morphological details. Okay, morphology gives us a visual identity, but the real story of impact is in the pathogenesis. Let's focus first on the classic, C. diphtheria, and the disease it causes. Diphtheria.
Right. Corynebacterium diphtheria is the definitive causative agent of diphtheria, primarily known as an acute illness affecting the upper respiratory tract.
Describe the typical presentation based on your source.
It often starts pretty mildly, actually. Symptoms like a sore throat and maybe a low-grade fever.
Yeah.
But the hallmark, the really distinctive and dangerous sign, is the formation of an adherent pseudomembrane.
Adherent pseudomembrane. Okay. Where does this membrane typically appear?
It forms on the mucous membranes, most commonly on the tonsils, the pharynx, and or the nose.
And this membrane isn't just an unpleasant symptom, it's the main source of danger, isn't it?
Absolutely. Your source emphasizes the critical risk. This tough, adherent membrane can extend downwards into the larynx and trachea, physically obstructing the airway and potentially leading to asphyxia. Suffocation, basically. This is why diphtheria requires urgent medical attention.
What does this pseudomembrane look and feel like? You said adherent.
Yeah, adherent means it's firmly attached and difficult to remove. If you try to scrape it off, it actually leaves a raw, bleeding surface beneath, which is a diagnostic sign in itself.
Ouch.
And it typically appears grayish or maybe greenish gray.
Beyond the respiratory form, are there other ways diphtheria can manifest?
Yes, the source mentions cutaneous diphtheria. This involves chronic, non-healing skin lesions, often covered by a grayish membrane or crust.
Skin lesions, too.
Yeah, and infections can also occur at other mucous membrane sites, like the external ear, the eye causing conjunctivitis, and the genital mucosa. While less common than the respiratory form, these extralaryngeal sites are noted. For you, the dental student, infections involving the nose, tonsils, pharynx, or even the eye and ear are certainly relevant to a comprehensive head and neck assessment.
Definitely. Does the infection always remain localized to the site of the membrane or lesion?
No, and this is a key point. While the bacteria itself tends to remain localized to the site of initial infection, it produces a powerful exotoxin.
The toxin, right?
And this toxin can diffuse throughout the body and cause systemic damage. Your source lists systemic effects involving the kidneys, the heart leading to cardiotoxicity, and the nervous system causing neurotoxicity.
Heart and nerve damage. That's serious.
Very serious. These effects can occur even after the local infection is treated and are responsible for much of the morbidity and mortality associated with diphtheria.
The source notes these systemic effects are less common with cutaneous diphtheria. Why might that be?
Well, the source doesn't explicitly state why, but it's generally thought that toxin produced in skin lesions might be less readily absorbed into the bloodstream and distributed systemically compared to toxin produced on the highly vascularized mucous membranes of the respiratory tract.
Okay. Less direct access to the bloodstream from the skin.
Exactly. The dose of toxin absorbed and the efficiency of its entry into circulation likely differ depending on the infection site.
Here's where it gets really interesting, and it's the core mechanism of C. diphtheriae pathogenicity, that powerful exotoxin.
This toxin is the primary driver of the severe systemic effects. It's a polypeptide toxin that diffuses from the site of bacterial growth into the surrounding tissues and then enters the bloodstream to be carried to distant organs.
And its mechanism of action is quite fundamental to cell function, isn't it?
It is. The diphtheria toxin works by inhibiting protein synthesis in eukaryotic cells.
Inhibiting protein synthesis. How?
It does this by modifying a factor essential for the elongation of polypeptide chains during protein synthesis. Essentially, it shuts down the cell's ability to make the proteins it needs to survive and function. It grinds the factory to a halt.
Wow, inhibiting protein synthesis. That's devastating for any cell. What does that mean specifically for organs like the heart and nerves?
It means their cells can't repair themselves, can't produce essential enzymes, or carry out their normal metabolic processes. Damage to heart muscle cells due to inhibited protein synthesis can lead to myocarditis and heart failure.
Myocarditis. Inflammation of the heart muscle.
Exactly. And damage to nerve cells can lead to paralysis, affecting breathing, swallowing, or limb movement. The severity of these effects depends on the amount of toxin absorbed and which organs are hit hardest.
And the ability of C. diphtheriae to produce this toxin isn't always guaranteed, is it? The source mentions a genetic aspect.
That's correct. Toxin production is not an inherent trait of all C. diphtheria strains. The gene encoding the diphtheria toxin, it's called TOX, is typically carried on a bacteriophage. That's a virus that infects bacteria.
A virus gives the bacteria the toxin gene?
Pretty much. If a non-toxigenic strain of C. diphtheria gets infected by a specific type of bacteriophage, a lysogenic one carrying this TOX gene, the strain can become toxigenic. It's called lysogenic conversion.
Lysogenic conversion, okay.
Your source mentions plasmids might encode it too, and plasmids can carry toxin genes in other bacteria, but the classic mechanism for diphtheria toxin is this phage conversion. The key takeaway is that the toxin gene is mobile, it can be acquired.
So not every C. diphtheria is dangerous, only the ones that have acquired this TOX gene.
Precisely. Which is why testing for toxin production is so critical, as we'll discuss later.
And this toxin is also the basis for our highly effective diphtheria vaccine.
Exactly. The diphtheria vaccine is a toxoid vaccine.
What exactly does that mean, a toxoid?
A toxoid is a bacterial toxin that has been treated usually with formaldehyde to eliminate its toxic properties while retaining its antigenicity.
Antigenicity, meaning the immune system still recognizes it.
Exactly. It's like a detoxified version of the toxin. It's harmless, but it still looks like the real toxin to your immune system.
So when you're vaccinated with the diphtheria toxoid, your immune system sees it as foreign.
It mounts an immune response, producing antibodies specifically directed against the diphtheria toxin.
And why does vaccinating against the toxin rather than the bacterium itself protect you from the disease?
Because the severe disease manifestations, the systemic effects on the heart, nerves, and kidneys are primarily caused by the toxin, not the bacteria directly invading those distant tissues.
Oh, okay.
So, by having antibodies ready to neutralize any circulating toxin produced by an infecting strain of C. diphtheriae, your body can prevent that toxin from reaching and damaging your vital organs, thereby preventing the severe systemic form of diphtheria. The vaccine protects against the effects of the toxin, which is the main threat.
That's a powerful concept, fighting the weapon rather than just the wielder. So that's the big story of C. diphtheria, but your source makes it very clear that the Corynebacterium genus includes many other species that can cause disease, often referred to collectively as non-diphtherial Corynebacteria or just diphtheroids when discussing their morphology.
Yes, and the source highlights a critical characteristic of many of these other species. They are often opportunistic pathogens.
And in this context, opportunistic means?
They don't typically cause infection in healthy individuals with intact immune systems. They seize an opportunity, which usually means a compromised host.
And the source specifically calls out immunocompromised hosts as a key context. For you, the dental student, who might these immunocompromised patients be that you encounter in your practice?
Oh, you'll definitely see them. These could be patients undergoing chemotherapy or radiation therapy for cancer, patients with HIV/AIDS, transplant recipients on immunosuppressant drugs, individuals with autoimmune diseases being treated with biologics or steroids, patients with poorly controlled diabetes, chronic kidney disease, or simply the very elderly or very young with less robust immune function.
Lots of different conditions.
Yeah. You will absolutely see these patients in a dental clinic, and understanding their increased susceptibility to opportunistic infections is paramount.
Okay, let's go through the other pathogenic species your source lists and briefly touch on their relevance.
First up, Corynebacterium ulcerans. The source notes it can cause infections that look very much like diphtheria, affecting the throat and skin.
Like diphtheria, so pseudomembranes.
It can, yeah. And importantly, some strains of C. ulcerans may produce the same diphtheria toxin as C. diphtheria.
Whoa, so a diphtheroid, that might actually make the diphtheria toxin. That's clinically important to differentiate.
Exactly. The source also notes its isolation from raw milk and cattle mastitis, suggesting a potential zoonotic transmission route from animals.
Interesting. Okay, next.
Corynebacterium jiceum. This is often referred to as group JK Corynebacteria. Your source provides significant detail on this one. It notes it frequently colonizes the skin, especially in patients with prolonged hospitalization, those who are neutropenic.
Neutropenic, low white blood cell count, right?
Yeah.
Common in chemo patients.
That's right. Or those receiving long-term antibiotics, which can disrupt normal flora. These are classic high-risk patient populations.
Definitely.
And C. jiceum can cause serious infections in these vulnerable patients, including septicemia bloodstream infections, foreign body infections associated with medical devices like intravenous catheters, prosthetic joints, or other implants.
Implants. Like dental implants, too, potentially.
Any foreign body, yeah. And critically, endocarditis, which is an infection of the heart valves.
Endocarditis again. This is a recurring theme and highly relevant to dentistry.
It truly is. And a critical point highlighted in your source about C. jiceum is its frequent and often extensive resistance to common antibiotics.
Highly resistant. That's a major problem.
It really is. This makes treating infections caused by C. jiceum particularly challenging and requires careful selection of antibiotics based on susceptibility testing. You can't just guess with this one.
That's a major clinical challenge for managing these infections. What other species are listed?
Corynebacterium ureolyticum, also known as group D2. This species is primarily associated with urinary tract infections.
Okay, urinary tract. And Corynebacterium pseudotuberculosis.
This one is interesting because it's typically an animal pathogen, causing things like caseous lymphadenitis in sheep and goats.
From animals again?
Yeah, but it can cause disease in humans, especially in immunocompromised individuals or those exposed to infected animals. Your source links it to endocarditis, both on native and prosthetic heart valves, pneumonia, lung abscesses, tracheobronchitis, and suppurative lymphadenitis infected and swollen lymph nodes.
Suppurative lymphadenitis, pus-filled lymph nodes. That's certainly relevant in the head and neck region, which you will be examining.
Definitely. Another opportunistic one with potential head and neck involvement.
Okay, what else?
Then there's Corynebacterium hemolyticum. This is notable because it's a recognized cause of pharyngitis at the sore throat.
Pharyngitis, like strep throat.
Similar symptoms, yeah. Your source provides a specific statistic. It accounts for up to 10% of pharyngitis cases in patients aged 10 to 30 years old. It can also cause an erythrogenic rash, similar to the rash seen with scarlet fever caused by certain Streptococcus species.
10% in that age range is actually a significant number for a dental student to be aware of when considering differential diagnoses for a patient presenting with throat pain. It's not always strep.
Absolutely. Good point. And finally, two species that are largely considered normal inhabitants but can occasionally cause problems. Corynebacterium xerosis and Corynebacterium pseudodiphtheriticum.
Xerosis and pseudodiphtheriticum.
These are found on the skin, conjunctiva, the membrane covering the eye and eyelid, and throat. Your source notes they can be occasional opportunistic pathogens, again, primarily in compromised patients, causing things like endocarditis in those with prosthetic heart valves.
So we see this pattern emerge again. Many of these other coryneiforms are commensals or environmental organisms that become problematic, specifically in vulnerable patients, with endocarditis being a recurring serious outcome.
Precisely. Understanding that these organisms are out there, often as part of normal flora on other body sites, and that they can cause serious life-threatening infections, especially endocarditis, in compromised patients, is vital for your practice.
How so?
Well, it informs your medical history taking, your risk assessment, and your decisions regarding patient management, including things like antibiotic prophylaxis for certain procedures in at-risk individuals. You need to know who's vulnerable.
Okay, so we know what they are, what they look like, and what diseases they can cause. How do we actually confirm their presence and identify them in the lab? Your source provides a good overview of laboratory diagnosis, relying on microscopy, culture, and virulence identification tests.
Yeah, this is a very practical section for understanding how a diagnosis is made. It starts, of course, with proper specimen collection. For suspected respiratory diphtheria or other lesions, swabs from the nose, throat, or the specific lesion should be obtained.
And a critical instruction from the source is...
collect these samples before initiating antibiotic therapy.
Right. Why is getting the sample before antibiotics so important?
Simple. Antibiotics are designed to kill or inhibit bacterial growth. If you start antibiotics before collecting the sample, you might kill enough of the bacteria to make them undetectable by microscopy, or, more commonly, prevent them from growing in culture in the lab.
Which leads to false negatives.
Exactly. False negative results, which can delay or prevent an accurate diagnosis and appropriate treatment.
Makes sense. Your source also mentions oral rinse samples.
It does, but clarifies this is for evaluating oral candidal carriage, looking for yeast like Candida albicans. That's a different organism entirely, but listed within the context of sample collection for oral or head and neck issues.
Got it. Note, specifically for your field, the source details collection for pus samples from dental abscesses.
Yes, and this is a crucial practical point for you. The source instructs that pus samples from dental abscesses should be collected by needle aspiration or strict aseptic swabbing.
Why the emphasis on needle aspiration or strict aseptic swabbing for a dental abscess? Why not just any swab?
Because dental abscesses, particularly those of odontogenic origin starting from the tooth, are almost always polymicrobial infections.
Polymicrobial. Many different bacteria involved.
Exactly. They contain a complex mix of bacteria, both aerobes and anaerobes, many of which are part of the normal oral flora just hanging out on the surface.
Yeah.
Using a needle aspiration or swabbing very carefully after surface disinfection to avoid contaminating the swab with surrounding mucosal or skin bacteria helps ensure that the sample collected truly represents the microbes within the abscess.
Not just the bystanders on the outside.
Precisely. It helps identify the actual pathogens driving the infection. Contaminated samples can lead to really misleading lab results, maybe suggesting treatment for the wrong bug.
Okay, aseptic technique is key for abscess samples. What happens next in the lab? Microscopy.
Yep, microscopy is often the first step. Direct smears, stained with gram stain or maybe alkaline methylene blue, can be examined for the presence of gram-positive rods and those characteristic arrangements, V-shapes, palisades, or even the Chinese letter pattern.
But we noted earlier that morphology alone isn't sufficient for a definitive diagnosis of C. diphtheriae because of the similarity to diphtheroids.
Exactly. The source reiterates this point. While microscopy gives you a preliminary idea, okay, it looks like some kind of gram-positive rod here, you cannot definitively distinguish toxigenic C. diphtheriae from non-pathogenic diphtheroids based on gram stain alone.
This is where those special granule stains come back in.
You got it. The source mentions Albert, Neisser, and Ponder stains specifically for highlighting the metachromatic granules, which are more abundant and distinct in C. diphtheria compared to most diphtheroids. Seeing those beaded rods with granules is a strong indicator that warrants further testing.
Strong indicator, but not proof yet.
Right. The source also mentions immunofluorescence as a potential method for detecting toxigenic C. diphtheriae directly in smears. This technique uses fluorescently tagged antibodies that bind specifically to diphtheria bacteria expressing the toxin. It might be faster, but still often considered preliminary.
Okay. And again, the source notes using microscopy for Candida too.
Yeah, just reinforcing that a lab examining a sample from the oral region might be looking for fungi like Candida albicans as well as bacteria, often using gram stain for that too.
So microscopy gives us clues, but culture is usually necessary for definitive identification and, importantly, susceptibility testing. What are the growth requirements for C. diphtheria?
C. diphtheriae is aerobic or facultative anaerobic, grows best aerobically at 37 degrees C, body temperature, with a slightly alkaline pH of 7.2 to 7.4. It's a bit picky, needs enriched media for optimal growth, things like blood agar, serum-enriched media, or media containing egg.
Enriched media. And are there specific selective media used to help isolate it?
Yes, selective media are really helpful because clinical samples often have lots of other bacteria. Blood tellurite agar is specifically mentioned. The tellurite salts in the media inhibit the growth of many other bacteria, allowing Corynebacterium species to grow more easily.
Tellurite agar, and what do they look like on it?
After incubation for about 48 hours at 35 degrees C, Corynebacterium species produce distinctive gray to black colonies. This is because they reduce the tellurite chemical in the agar. The appearance of these colonies can also vary slightly, depending on the biotype of C. Diphtheria mitis, intermedius, gravis.
Biotypes, different subtypes of C. diphtheria. Okay. What about on regular blood agar?
On blood agar, C. diphtheria can show variable patterns of hemolysis. That's the breakdown of red blood cells around the colony. Depends on the biotype again. Mitis strains often show beta hemolysis, complete clearing. Intermediate strains are non-hemolytic, and gravis strains are usually non-hemolytic or maybe weakly hemolytic.
And in liquid media, like broth?
In broth, they can grow as a pellicle, a film on the surface, or as granular deposits at the bottom.
Do the other Corynebacteria, the diphtheroids, have similar growth requirements? Are they as picky?
Generally, no. The source notes that. In contrast to C. diphtheria's need for enriched media, many other Corynebacteria diphtheroids grow just fine on ordinary laboratory media. That's another point of differentiation in the lab.
Easier to grow. Now, going back to dental abscess samples, your source mentions specific incubation conditions for these.
Yes, for samples from dental abscesses, the instruction is incubation on blood agar plates for 2 to 3 days at 37 degrees C.
Why the longer incubation time, up to 3 days? That seems longer than for many routine cultures.
That's a key piece of context provided by your source, really important for you to understand. It states that this extended incubation period is used because most oral pathogens found in abscesses are slow-growing anaerobes.
Ah, okay. So even though Corynebacterium isn't strictly anaerobic.
Right. While Corynebacterium itself is primarily aerobic or facultative anaerobic, the lab protocol for a dental abscess sample is designed to recover the full spectrum of likely culprits. This includes many strict anaerobic bacteria that just take longer to grow.
So the lab is hedging its bets, making sure they don't miss the slow-growing anaerobes that are common in these infections.
Exactly. The incubation conditions are optimized for the most common, potentially fastidious organisms encountered in dental abscesses, ensuring that even slower-growing anaerobes or facultative organisms like Corynebacterium are detected if present.
That makes sense. The lab is covering all the bases for a typical polymicrobial dental infection. Once you have a culture, you need to identify the specific species. How's that done?
That's where biochemical tests come in. These tests assess the metabolic capabilities of the bacteria, like what sugars can it ferment? What enzymes does it produce? These reactions help differentiate between species within the Corynebacterium genus.
What are some key biochemical tests for C. diphtheria?
For C. diphtheria, important tests include fermentation of specific sugars like glucose, galactose, maltose, and dextrin. The source also mentions testing for cystinase activity.
And how do these tests help distinguish C. diphtheria from diphtheroids?
A key differentiating point mentioned in the source is sugar fermentation patterns. Diphtheroids are often positive for fermentation tests that C. diphtheria is negative for. For example, many diphtheroids ferment sucrose and mannitol, whereas C. diphtheria typically does not. These differential fermentation patterns are crucial for species identification.
So different metabolic fingerprints.
Exactly. And of course, commercial identification kits are also widely used in modern labs for rapid biochemical profiling. They have panels of tests ready to go.
So even if something looks like Corynebacterium under the microscope and grows on selective media, you need biochemical tests to confirm the species. But if you suspect diphtheria caused by C. diphtheria, confirming a species isn't enough, is it? You need to know if it's producing the toxin.
Absolutely. Toxogenicity testing is essential for diagnosing toxigenic diphtheria because, as we said, not all strains of C. diphtheria are toxigenic. These tests demonstrate whether the isolated strain is capable of producing the diphtheria exotoxin.
What methods are used for toxogenicity testing according to the source? Are they still using guinea pigs?
Well, the source mentions those older historical methods like in vivo tests involving subcutaneous or intracutaneous inoculation of guinea pigs to see if they develop a reaction. Toxin is lethal to them. But yeah, modern labs rely much more on in vitro methods now.
Safer and faster, presumably. Tell us about the in vitro tests.
The classic in vitro test is the ELEX gel precipitation test.
ELEX test. How does that work?
It involves inoculating the Corynebacterium isolate onto a specific agar medium that has a strip of paper soaked in diphtheria antitoxin embedded in it. If the isolate produces a toxin, the toxin diffuses out from the bacteria and the antitoxin diffuses out from the strip. Where they meet in the agar at the right concentration, they form visible lines of precipitate. A positive result.
Lines in the gel.
Other methods include tissue culture tests, where you look for the toxin cytotoxic effect, its ability to kill eukaryotic cells, like monkey kidney cells in a dish, and enzyme-linked immunosorbent assays, ELISIS, which can directly detect the toxin protein itself using antibodies.
And the most modern approach involves molecular biology, PCR.
Yes. Polymerase chain reaction, or PCR. The source notes that PCR is a very useful method for diagnosing diphtheria.
What's fascinating here is how PCR works in this context. It's not looking for the bacteria itself or the protein.
Right. Instead of culturing the bacteria or detecting the toxin protein, PCR directly detects the presence of the diphtherial toxin gene, known as TOX, in the clinical specimen.
Why is detecting the gene so powerful, particularly in a clinical scenario?
Well, it allows for rapid detection, which is great, but critically, it can detect the potential for toxin production even from non-viable organisms.
Non-viable, like dead bacteria.
Exactly. For example, if antibiotic therapy was started based on clinical suspicion before definitive lab results were available, which often happens with suspected diphtheria, the bacteria in the sample might be killed or inhibited, making culture difficult or impossible.
But the DNA is still there.
The bacterial DNA containing the TOX gene can still be detected by PCR in the original clinical specimen. So it can provide a diagnosis of toxigenic diphtheria even after treatment has begun. This is incredibly valuable for confirming the diagnosis and guiding public health measures.
That is a big deal. Wow. The source also briefly mentions other antigen detection methods.
Yeah, it just throws in examples like direct fluorescent antibody, DFA, tests and direct enzyme immunoassays, EIA, in the context of detecting gonococcal antigens, just as examples of other rapid detection techniques. But it clarifies they are specific to Corynebacterium based on the material provided here, just illustrating the types of tests labs use.
Okay, so understanding this whole diagnostic pathway is essential. Bringing it back to you, the dental student, why is knowing these lab specifics important? You're not running the tests yourself.
No, but it's vital for interpreting lab reports you receive. When you submit a sample from a dental abscess, knowing it's incubated for 2-3 days to catch anaerobes, and that different tests are used to distinguish a harmless diphtheroid from a more concerning organism like C. gichium, informs your understanding of the results.
Right. It helps make sense of the report.
Exactly. Knowing that specialized stains are used for granules, that biochemical tests differentiate species, and that PCR can confirm toxin potential quickly provides context for the diagnostic process for relevant organisms. And maybe most importantly, understanding proper sample collection, like strict aseptic technique for an abscess, directly impacts the quality and accuracy of those lab results you rely on. Garbage in, garbage out.
Absolutely. Okay, once we have a diagnosis, the next step is treatment. Your source outlines the approach to fighting these infections.
For diphtheria caused by C. diphtheriae, the source stresses that treatment must be initiated immediately based on clinical suspicion. You cannot wait for lab confirmation.
Why the urgency?
It's a medical emergency because of the rapidly spreading toxin and the risk of airway obstruction. Time is critical.
And treatment involves two main components.
Yes, antitoxin administration and antibiotics.
Why do you need both antitoxin and antibiotics? Why not just kill the bacteria?
Good question. They serve different crucial purposes. The antibiotics, typically penicillin or erythromycin according to the source, are given to kill the bacteria. This stops them from producing more toxin and helps clear the infection at the source, like the throat membrane.
Okay, stop the factory.
Right. But the toxin that has already been released and has diffused into the bloodstream needs to be neutralized before it does more damage. That's where the antitoxin comes in. Diphtherial antitoxin is a preparation of antibodies, usually made in horses actually, that bind to and inactivate circulating toxin molecules.
So it mops up the existing toxin.
Exactly. It prevents the toxin from reaching and damaging target cells in organs like the heart and nerves. You absolutely need both. Antibiotics to eliminate the toxin factory, the bacteria, and antitoxin to neutralize the existing toxin that's already causing systemic damage.
Got it. What about for other coroniform infections caused by the opportunistic species we discussed, like C. jiceum?
For those, the treatment often also involves penicillin or erythromycin, but, and this is a big but, treatment really depends on the specific species and its susceptibility profile.
And here's that critical challenge again, highlighted by your source, antibiotic resistance.
Yes, particularly with Corynebacterium jiceum. The source explicitly states that C. jiceum can be very resistant to common antibiotics.
Very resistant.
Yeah, this is a significant clinical hurdle and emphasizes the need for performing antibiotic susceptibility testing on isolates from serious infections like septicemia or endocarditis caused by this organism. You have to know what it's susceptible to.
Otherwise, your treatment might not work at all.
Precisely. Knowing the resistance pattern is crucial for selecting an effective antibiotic. The source also briefly mentions that rhodococcus infections, a related genus, are often treated with vancomycin or maybe a combination like erythromycin plus rifampicin. Yeah. Again, just illustrating how resistance profiles dictate treatment choices for these less common but potentially difficult to treat pathogens.
So for a dental student, being aware that seemingly minor or commensal bacteria like these coroniforms can, in compromised patients, cause serious infections that are also difficult to treat due to resistance, that's a really important clinical consideration.
It certainly is. It impacts everything from your risk assessment before a procedure to your understanding of why a patient might require complex medical management, maybe involving infectious disease specialists for an infection that originated from an opportunistic organism they picked up. It underscores the complexity of managing medically compromised patients.
All right, we've built the picture. We've looked at the bacteria generally, their appearance, the main diseases, the other players, how to find them, and how to fight them. Now, let's pull it all together and explicitly connect it back to your field. This is the core section specifically for you, the dental student, the direct relevance of Corynebacterium and related concepts to dentistry.
Let's expand on every point the source makes here.
Okay.
First, the concept of normal oral flora. Your source reiterates that the oral cavity is home to many indigenous bacteria, including non-pathogenic Neisseria species and diphtheroids, those coroniform bacteria that morphologically resemble C. diphtheriae but are typically harmless residents.
What's fascinating here, and something specific to the oral cavity, is that the source names a single species as the only true coroniform organism found as a normal inhabitant.
Yes, Corynebacterium matricoti.
Matricoti.
It's described as a rather unique morphology, resembling a whip. This specific species is consistently found in the healthy oral microbiome.
A whip-like morphology. That's quite distinct. How does understanding this specific oral coroniform and the general concept of normal flora benefit a dental student?
Well, understanding the composition of normal oral flora is fundamental, isn't it? These bacteria occupy ecological niches, they compete with potential pathogens, they contribute to oral health homeostasis. Knowing that C. matricoti is a normal resident means finding it in a sample from the mouth might be expected and doesn't necessarily indicate infection.
Unless the context changes.
Right. Unless the context changes, which we'll get to. It helps to find the baseline of a healthy oral environment, knowing who's supposed to be there.
Speaking of context changing, that leads us back to the concept of opportunistic infections, which is hugely relevant for dentistry.
Absolutely. The source reiterates that diphtheroids, these coroniform bacteria other than C. diphtheria, can act as opportunistic pathogens.
And it emphasizes the context in compromised patients.
Exactly. As a dentist, You will see patients who are immunocompromised due to systemic disease, medical treatments, or age. Understanding that bacteria that are normally harmless residents of the skin, throat, or other body sites, or even the mouth itself, can cause serious infections when a patient's immune system is suppressed, is absolutely vital for risk assessment and treatment planning.
What kind of opportunistic infections originating from or relevant to the head and neck region might occur in these patients?
Well, it could range from localized infections if the oral mucosa is compromised, like in cases of mucositis from cancer therapy. Or it could be more severe systemic infections. Given that these opportunistic corniforms are normal flora of the skin and upper respiratory tract, infections could manifest as skin or soft tissue infections in the head and neck area.
Okay.
Or, as we've discussed, they could potentially spread systemically to cause bloodstream infections or endocarditis. A dentist needs to be aware of the potential for unusual infections in these patients and the importance of collaborating with their medical team. You might spot something that needs medical follow-up.
Let's circle back to diphtheria and its oral manifestations. While the severe form is respiratory, the pseudomembrane can appear on the tonsils and oropharynx.
Yes, and this is a direct point of relevance for you. During a routine oral and pharyngeal examination, a dentist or dental student might be the first healthcare professional to visually identify the characteristic grayish, adherent pseudomembrane covering the tonsils or the back of the throat.
You might actually see it.
You might. And recognizing this as a potential sign of diphtheria is critical because, as we discussed, it's a medical emergency requiring immediate action, including antitoxin administration. The ability to spot this non-dental but orally visible manifestation is a key part of being a comprehensive healthcare provider. You're looking at the whole picture.
Now let's get right into dentoalveolar infections, the infections within the tooth and surrounding structures. The source makes a specific critical point about Corynebacterium matrichodia here.
This is highly relevant, yeah. Your source explicitly states that Corynebacterium matrichodii, that unique whip-like normal oral commensal we just talked about, has been isolated from miscellaneous cultival bacteria in primary endodontic infections.
Isolated from primary endodontic infections. That means infections within the root canal system of a tooth that hasn't been treated before, deep inside the tooth.
Exactly. The source notes its presence among the complex mix of bacteria that are cultured from these deep infections.
What's the implication of finding C. matrichodi, a normal resident of the healthy oral cavity, in a primary root canal infection? Is it causing the infection?
Well, the source just states its isolation, indicating it is present in these polymicrobial communities within the infected tooth. It doesn't explicitly define its specific role, whether it's a primary pathogen initiating the infection, or maybe a secondary opportunist joining an existing infection, or perhaps simply a passive participant from the diverse oral flora that finds its way into the root canal space and survives there.
So we don't know its exact role from this source, but it's there.
It's there. Its presence tells us that this organism, normally considered harmless, can infiltrate and be part of the bacterial population in deep pathological spaces, like an infected root canal system. This really underscores the polymicrobial and often complex nature of endodontic infections. They can involve a wide range of bacteria, including those typically thought of as benign commensals, just taking advantage of the situation.
Moving to systemic infections of dental relevance. This is perhaps the most significant connection mentioned in your source, particularly endocarditis.
This link between oral bacteria and infective endocarditis is a major area of concern in clinical guideline development in dentistry. It's something you absolutely need to know about. Your source mentions C. jichium again in this context, noting its ability to cause endocarditis specifically in immunocompromised hosts.
C. jichium and endocarditis in the immunocompromised. And the source also brings in another organism here to emphasize the point, Corynebacterium hominis.
Yes, and it's important to note, as the source does, that Corynebacterium hominis is a gram-negative bacillus, not a Corynebacterium. It's a different type of bacteria altogether.
So why mention it here?
It's included in this discussion because it is another example of an oral commensal organism, part of the normal mouth flora, that is a recognized cause of endocarditis, particularly in patients with pre-existing cardiovascular disease, such as damaged heart valves.
Why is it so important for the source to juxtapose C. jichium, a gram-positive opportunist, and Corynebacterium hominis, a gram-negative commensal, in the context of endocarditis for a dental student?
This juxtaposition powerfully reinforces the concept that the oral cavity is a reservoir for a variety of bacteria, both gram-positive opportunistic coryneiforms like C. giacinum and gram-negative oral commensals like Corynebacterium mohammadis that have a potential to cause serious systemic infections, especially endocarditis.
So the mouth harbors multiple potential threats for endocarditis.
Exactly. Many dental procedures, even things like routine cleaning or extractions, can cause a transient bacteremia.
Bacteria getting into the bloodstream.
Right. Usually harmlessly cleared by the immune system. But in patients with certain predisposing heart conditions like prosthetic valves, a history of previous endocarditis, or certain congenital heart defects, these bacteria circulating in the blood can adhere to and infect the damaged heart valves or prosthetic material, leading to infective endocarditis.
Which can be fatal.
It can be. Understanding this risk is fundamental for identifying patients who require antibiotic prophylaxis, taking antibiotics before certain dental procedures to prevent this serious complication. It highlights how localized oral flora can have profound systemic consequences. What happens in the mouth doesn't always stay in the mouth.
That's a critical takeaway. Another systemic connection relevant to the head and neck region is pharyngitis caused by C. hemolyticum.
Right, as we noted earlier, it's a significant cause of sore throats in that 10-30 age range. While not a dental infection per se, patients may present to a dental clinic complaining of sore throat that they might attribute to a tooth issue, or maybe they just see you first.
So it's part of the differential diagnosis for throat pain.
Exactly. Knowing that C. hemolyticum is a potential cause expands your differential diagnosis beyond just Streptococcus pyogenes, strep throat, or viral infections, allowing for more comprehensive patient assessment and appropriate referral if needed.
Moving to cross-infection control, a paramount concern in any healthcare setting, including dentistry. Your source mentions the DTP vaccine in this context.
The diphtheria tetanus pertussis, DTP vaccine, or similar boosters like Tdap, is noted as important for dental healthcare workers.
Why specifically for dental professionals is the risk of diphtheria high?
It's more about personal protection, however low the baseline risk is. While diphtheria is rare in vaccinated populations, dental professionals work in very close proximity to patients' airways and handle instruments that can generate aerosols containing respiratory droplets. There is a theoretical risk, however small, of exposure to C. diphtheriae or toxigenic C. ulcerans carried by a patient, perhaps asymptomatically or unknowingly.
So better safe than sorry?
Pretty much. Being vaccinated protects the dental healthcare worker from potentially acquiring these infections in the workplace. It's part of occupational health and safety.
Makes sense. The source also includes a point about dental unit water lines, DUWLs, as a concern for biofilm formation and a potential source of cross-infection.
Yes, biofilms in the water lines. Definitely not a pleasant thought for infection control. Those thin tubes carrying water to your handpieces and air water syringe can harbor bacteria.
Are Corynebacterium a major problem in DUWLs, according to the source?
Actually, no. The source primarily links DUWL contamination to other pathogens, like Legionella species, which can cause Legionnaire's disease, a severe pneumonia, and non-tuberculous mycobacteria. It doesn't specifically finger Corynebacterium in this section.
So why is it mentioned here?
Its inclusion here, within the discussion of cross-infection control relevant to dentistry, serves to emphasize the broader importance of maintaining rigorous infection control protocols. This includes properly managing DUWLs according to guidelines, flushing, using appropriate disinfectants, monitoring water quality to minimize the transmission of any potential pathogen, whatever it might be, within the dental clinic environment. It's about the overall principle of water safety in the clinic.
Right, controlling biofilms generally. Finally, the source mentions oral manifestations in compromised patients more broadly, touching on mucositis.
It notes that patients who are immunocompromised, such as those undergoing radiotherapy or chemotherapy for cancer, often develop oral problems like mucositis. That's painful inflammation and ulceration of the oral lining, the mucosa.
Very common and debilitating side effect for those patients.
It is. And the source links this mucositis to changes in the oral microbiome.
Yes, it states that this mucositis may be associated with overgrowth or infection by gram-negative aerobes and facultative anaerobes, specifically mentioning Klebsiella species, which are related to the Enterobacteriaceae family common gut bacteria. Okay, again, Klebsiella is not Corynebacterium. Why is this point included in the section on Corynebacterium relevance? Seems a bit tangential.
I think it's included because it reinforces the overarching theme of this section. Compromised patients are susceptible to opportunistic infections by organisms that are either normal flora or colonizers from elsewhere. Even though the specific organism mentioned here is Klebsiella, The principle is the same as with opportunistic Corynepacterium or Corynebacterium.
The principle being?
The principle being that when host defenses are weakened, the normal balance of the oral microbiome is disrupted. This allows various opportunistic bacteria, gram-positive rods like Corynepacterium, gram-negative rods like Klebsiella, yeasts like Candida to proliferate and contribute to oral pathology like mucositis, or potentially become a source of systemic infection if they gain entry. This general susceptibility to a wide range of opportunistic pathogens is a crucial concept for you as a dental student treating medically complex patients. You need to expect the unexpected sometimes.
Wow, that really connects the dots, showing how understanding this seemingly broad genus Corynebacterium and the related concept it introduces like opportunism and the oral systemic link is far more relevant to daily dental practice than one might initially think. Definitely not just about diphtheria anymore.
Not at all. It's about recognizing the potential for normal flora to become pathogens in the right circumstances, understanding the specific risks in vulnerable patient populations, appreciating the profound oral systemic link, especially with endocarditis, and mastering the diagnostic and infection control principles that apply to a whole range of organisms you might encounter or need to consider.
Okay, let's synthesize this deep dive and pull out the key takeaways specifically for you, the dental student, based on the source material. What absolutely needs to stick?
Okay, first, internalize that Corynebacterium is a diverse genus extending far beyond C. diphtherio and diphtheria. It includes common commensals like C. matrichotes, specifically in the oral cavity, but also opportunistic pathogens that pose significant risks, particularly to the medically compromised patients you will definitely see in your chair.
Okay, diverse genus includes commensals and opportunists. Got it. Second,
remember that their appearance under the microscope is varied. The pleomorphism, the club shapes, the Chinese letters, and those distinct metachromatic granules. This visual complexity, combined with specific staining, culture requirements, and biochemical tests, makes accurate laboratory identification complex, but absolutely essential for guiding diagnosis and treatment, especially for distinguishing potentially dangerous pathogens from harmless residents that might look similar at first glance.
Morphology and lab ID are key but tricky. Third takeaway.
Third, recognize the specific species and conditions discussed that have direct relevance to your practice. Think of C. matrichote not just as normal oral flora, but also as something isolated from endodontic infections. Crucially, understand the significant link between various oral bacteria, including opportunistic corniforms like C. jiceum and oral commensals like Corynebacterium hominis, and the risk of infective endocarditis in susceptible patients undergoing dental procedures. Know about prophylaxis guidelines. And also be aware of C. hemolyticum as a potential cause of pharyngitis when doing a differential diagnosis.
Specific oral links: endodontic infections, endocarditis risk, pharyngitis. Okay. And finally,
perhaps the most important clinical takeaway. Understanding the increased susceptibility of compromised patients to opportunistic infections, whether those infections are oral or systemic by these types of bacteria. And coupled with that, maintaining rigorous infection control practices, including your own personal vaccination and proper waterline management, are paramount for ensuring the safety and well-being of both your patients and yourself. Patient safety and provider safety.
Understanding risk in compromised patients and rock-solid infection control. Got it.
This deep dive into Corynebacterium really highlights the interconnectedness of oral health with overall systemic health. It demonstrates how even seemingly simple bacteria can have hidden potential and complex implications that are vital for you to understand as you move forward in your dental career.
It absolutely does. We've explored this fascinating genus and its connections to dentistry using your provided source material, moving from the microscopic details right through to the critical implications in your field.
Yeah, understanding this broader microbial landscape, the potential roles of organisms found inside and outside the oral cavity, is truly essential for providing comprehensive, safe, and effective patient care. It's more than just drilling and filling.
Definitely. So here's a provocative thought to leave you with, something to chew on. Given the diverse nature of these bacteria, we've seen harmless residents like C. matrichote in the healthy mouth, but also serious opportunists found even in root canals or infecting heart valves. How does appreciating the entire microbial landscape of the body, not just focusing solely on the organisms traditionally linked only to teeth and gums, how does that change how you think about assessing risk, taking a medical history, and planning treatment for all your patients, especially those with complex medical histories?
It's a lot to consider, but really crucial as you develop your clinical perspective and judgment.
Thanks for joining us for this deep dive.